Hi, my name is Allure, and over the summer I worked in Dr. Roepke's lab virtually, working on cognitive health of ovary-intact postmenopausal mice fed high-fat diets. It is known that in menopause, the decrease in estrogen production along with the metabolism of estrogens in the gut may impact cognitive health. In Roepke's lab, we used a chemical-induced ovarian-intact menopausal model. We did so by injecting 4-vinyl-cycloxane diepoxide, also known as VCD. As for our control mice, they were injected with oil. It was hypothesized that the coconut-saturated fatty acid diet would increase cognitive health, while the omega-6 polyunsaturated fatty acid diet would result in a cognitive decline. The methods were the use of 3 behavioral tests and 1 blood pressure system test. To begin with the behavioral test, Y mains was one of them. This is set up as a spatial navigation. This is a single day of testing, and it is a 15-minute test. The test consists of 5 minutes of limited exploration, 5 minutes of purgatory cage, and 5 minutes full exploration. This test is used to assess short-term memory in mice, as well as curiosity. Then there is VCD-SOAR, also known as spatial object recognition test. And this behavioral test consists of 4 days of testing. On day 1, there is a 5-minute recorded exploration. On day 2 and 3, there's a 5-minute non-recorded exploration. And for day 4, test day, there's a 15-minute test. 5 minutes exploration, 5 minutes purgatory cage, 5 minutes exploration. As for VCD-NOR, this is novel object recognition. This behavioral test consists of 5 days of testing. On days 1 through 4, there's a 5-minute non-recorded exploration. On day 5, which is test day, there's a 15-minute test. 5 minutes exploration, 5 minutes purgatory cage, 5 minutes exploration. As for all 3 of these behavioral tests, usable data is generated same day, and it takes about 3 to 5 minutes to clean up and prep for the next mouse. Moving on to VCD CODA. CODA is a machine. It's a non-invasive blood pressure system that accurately measures the 4 blood pressure parameters in the tail of the mice. This is a test used for testing the mice for their cardiovascular and capillary health by putting the animals through a machine called the CODA. Blood pressure is assessed for sake of the fed diet to the mice and its relation to cognitive function. In terms of results, if we take a look at figure 1, which is the VCD CODA data, you will see that the CODA results favored the hypothesis. The oil/control group experienced low pressures than the VCD injected group. As for figure 2, which is the VCD-SOAR data, it can be seen that the spatial object recognition hypothesis was supported and rejected, being as though the oil/control group accepted the hypothesis while the VCD injected group rejected the hypothesis. Again, the hypothesis being that the coconut-saturated fatty acid diets will improve cognitive health, while omega-6 polyunsaturated fatty acid diets will decline cognitive health. As for figure 3, it can be seen that the hypothesis was rejected for both the oil/control group and the VCD group. Then in figure 4, there was no statistical significance, but a trend was showed towards the VCD injected group having better cognitive health. So overall, the hypothesis was rejected and accepted in multiple scenarios of the testing, as far as the behavioral testing and the blood pressure testing. As for the future directions of the Roepke lab, metabolic phenotyping would be the next steps on the following, as well as gut microbiome analysis. To end things off, I would like to acknowledge and thank the Douglas Project Super Summer Stipend Research Program, as well as my faculty mentor, Dr. Roepke, for allowing me to attend his lab over the summer and over the spring semester, as well as my lab manager Ali Yazrebi, who was also my mentor for the summer, helped me with Excel and other things as well to succeed in the summer program. And I would also like to thank the lab members for their guidance and honor and patience, as well as everyone else. Thank you.